This is Protein Synthesis by Sam and Luke.
The building blocks of DNA are called nucleotides. These nucleotides are made of three ingredients: phosphates, pentose sugars, and nitrogen bases. The actual nucleotide looks a bit like an L. The four nitrogen bases are called adenine, thymine, guanine, and cytosine. Guanine and cytosine always form a pair, and adenine and thymine always form a pair. Guanine and cytosine are bonded together with 3 hydrogen bonds, while thymine and adenine are bonded with only 2. Nucleotides face each other and attach to form a sort of ladder shape that, when twisted, forms a shape called a double helix. This is arguably the most important shape in the world.
The first process of protein synthesis is transcription. This is when the DNA is copied into mRNA. This whole process happens in the nucleus because that is where the DNA is located. So, who does the copying of the DNA? The answer is RNA polymerase. RNA polymerase works by bringing in nucleotides to match the DNA. In this example, it would bring in a C, U, and a C. After this, mRNA is created. After the whole process of transcription is over, the mRNA will leave the nucleus.
The next step in protein synthesis is called translation. When the mRNA exits the nucleus, it goes into an organelle called the ribosome. This is called tRNA. It acts as a sort of shuttle bus for amino acids. Every 3 bases on mRNA are called codons. These also exist on tRNA and are called anticodons. Every codon on the mRNA will match up with an anticodon on the tRNA, which brings with it a unique amino acid. As this process continues, amino acids begin to form chains called polypeptide chains. These eventually fold up to create the final protein.
The next step of protein synthesis is folding amino acid chains. The first structure is called primary structure. This is basically a super long chain of amino acids. The second structure is called secondary structure. The amino acids are folded in a repeating pattern because of the hydrogen bonding of the peptide backbone. The third structure is called tertiary structure. This is a three-dimensional folding pattern due to side chain reactions. The black lines that you see in the drawing are the pleated sheets in the protein. The last and final structure of the protein folding process is quaternary structure. This is basically a protein with more than one amino acid chains. So, what is the importance of proteins? Why do we need proteins? Proteins play really important roles in the body. For example, it controls circulation, immunity, and communication between cells. It also catalyzes chemical reactions. Without proteins, you would not be able to survive.